We're at the IAS meeting in Vancouver. We're here with Sharon Lewin, the director of Peter Doherty Institute for Infection and Immunity at the University of Melbourne. And you have been so kind to do these interviews with us over the years. And this time you're going to do something which is really important for our audience because it is kind of telling us about what the future will look like. Maybe a little bit what the virus, how the cure will maybe look like. Because it keeps on changing in our minds, doesn't it?
Yeah. I think I can really understand why there's some confusion around that. As you and many of your viewers know, there's been a real big, renewed scientific effort and enthusiasm around finding a cure. But a cure means different things to different people. And the long-term aspirational goal is really to find a cure and eliminate HIV.
But I think that when we were here the last time in the conference in '96, where we thought that the drugs were going to be sufficient to give us a cure.
Yeah.
And so that was our cure in our minds then.
Exactly.
Which is very different than what we're looking at today.
Exactly. Well, actually back in '96, when antiretroviral therapy first came along and potently suppressed virus and virus disappeared from blood, people really did think that was going to be a cure and that maybe you needed to be on treatment for 3 years and the virus would decay.
And then it was 10 and then maybe 99 years.
Yeah. But actually, if you think back, within months of that discovery around antiretroviral therapy, latency was discovered and identified, and that is the big barrier to a cure, these long-lived latently infected cells where the virus is able to get into the DNA and hide out in those long-lived cells. And so very rapidly it became apparent that antiretroviral therapy is not going to cure HIV. You stop the drugs, the virus comes straight back up. So what do we mean by cure in 2015? I think really the field is looking at trying to induce long-term remission, and remission is exactly as how you think about cancer, meaning that the virus is still there, it could potentially come back, but it's at very low levels and not causing any adverse effects on the person. We still do, and we used to call that functional cure, but I think that's confusing, and I think you'll find that many people in the field, and you'll see from this conference, are talking about remission. And that, if we could achieve remission, meaning people could safely stop treatment and the virus stay under control for years, that would be a very big advance. And then the next step would be a real true cure.
I was talking with Nicholas just a moment ago about the importance of, basically getting the people tested right away. I think we can't hit that message hard enough and often enough. Because the quicker you identify your HIV status, you can have a different viral set point and maybe be a better candidate for a cure of whatever it might be down the line. The less your virus is controlled or consumed into your body, the better off you are. So I like to give that message out. That means more testing, more frequent testing, depending upon the level of risk.
Yeah, there's a whole lot of benefits for starting treatment early. We heard at this meeting around the first formal presentation of START showing a clear benefit for if you treat when there's no immune damage compared to if there's some modest immune damage, and a survival benefit. And added to that, from a cure perspective, if you treat early, the amount of virus that goes into hiding is much, much less, and your immune system and capacity to recognise the virus that goes into hiding is much better if you start treatment early.
I know this would probably be a question for Steve, but as far as inflammation, is the inflammation proportionally
if you start earlier, inflammation is lower. In fact, there are some recent studies showing that if you start early, inflammation on treatment is almost at the levels of a uninfected person. So I think inflammation is really, we see more of it in people that start late.
That may be part of the reasons why we have this accelerated, if you call it that, accelerated aging.
And the inflammation is linked to these other adverse outcomes. If you start earlier, survival benefit, smaller reservoir, less inflammation. To start early means to know your status and get tested frequently, and hopefully with making testing easier, that's going to be possible to implement. At the moment, that's the real challenge, to get people tested.
Don't go into denial. Denial does nothing for anyone. Just face the reality. Face the reality if you're at a risk for being positive. Realize that and deal with it appropriately.
Yeah, getting tested and getting tested regularly, and staying, and seeking help
to make a big difference on the length of your life, your quality of life, and everything.
Yeah, again, the studies definitely show that. We heard that this morning, that now life expectancy for people living with HIV is the same. In fact, it might even be better than people without HIV.
Because they're having more attention, more medical attention.
Yeah,
I think that could be very true.
But the survival benefit is in people that start with a reasonable, with minimal damage to the immune system. There's if you wait for damage to the immune system, the life expectancy doesn't quite reach normal.
So some of the things that you look forward to with hope in the future in the cure work, in the cure research, what would some of those things be?
Well, maybe we should talk about what we might see in the next couple of years. And I think what we're going to see in the next couple of years is the results of a number of really important clinical trials. So we heard at this meeting about the remission in the French teenager, and another report of remission, the first report in a baby treated. But we don't still know how common this is post-treatment control, meaning you're on treatment for a short period of time, then stop and the virus is in control. How common it is, why do some people have the capacity to control the virus and others don't? And the best way to answer that is in a clinical trial of people who initiate treatment early, or babies who get treatment administered at the time of birth, on treatment for a while, and then stop treatment safely, monitor closely, and see how long it takes for the virus to come back. And those studies are happening now, certainly in adults who initiate a treatment early, and I think we'll see the results of those studies largely coming from Thailand in the next year or so. I think we're going to see some really interesting, small but important intervention studies of shock and kill. We've talked. There've been a number of studies presented on shock, waking up the virus, and a number, but many people recognising that shock alone doesn't eliminate the cell.
But you kind of have to do that work separately, and then put them together with some other.
Yeah,
I mean, the shock and kill is obviously.
There have been. I think we're not. I think the studies of combination shock and kill trials are happening, and they're either about to happen or happening, and we'll see the results of those.
But
soon.
that is one approach. There's mixing that with a vaccine, maybe something down the line that would make the whole thing happen, synergistic.
Well, when we talk about kill, we mean the vaccine, actually. Okay. Shock meaning waking up the virus with a drug like an histone deacetylase inhibitor, or one of the cancer drugs that activate virus. And the kill is usually
really
with a vaccine.
Okay.
And so these HDAC inhibitors plus vaccine studies are happening, one in Denmark and Germany in the US, and another in the UK. And perhaps another one soon coming out in Europe. So we know that HDAC inhibitors do shock the virus, wake up the virus, but the cell doesn't die.
But what I'm getting at is, would there be like a dual vaccine or a triple vaccine that might be more effective than just a single? And that.
The vaccine that is being chosen for these studies of shock and kill varies. Some of the vaccines are targeting antibodies, and some of them are targeting T cells. We don't yet know what the best kill in a shock and kill setting will be. But those studies are happening, so we're going to hear the results of those studies in the next 12 to 18 months. And there's some really important work being done on understanding how to measure persistent virus in people on treatment. There's a lot of different assays being developed. We don't yet haven't understood how those assays vary in different laboratories, what they're measuring.
And that was part of what you were getting at in the press conference, trying to standardize this within industry?
And standardize those assays, and most importantly, see if any of them predict time in remission. And again, that's a big study that's ongoing now, funded by the Gates Foundation.
So is there anything else that you want to bring out before we conclude?
Oh, I was going to. There are also some. Well, I think again, what's going to change the landscape a bit about is how we work. There's some really exciting new initiatives and partnerships between industry and academia. A great example of that is this new partnership between GSK and UNC. AmFAR have just announced $100 million towards the Cure initiative and are looking to establish a Cure Institute at a site in the US. They're also funding a range of grants to bring people who are non-HIV researchers into the field to try and address the problem. And I do think that maybe we will see some us benefiting from some of the advances in cancer immunotherapy that I think are very relevant to the cure agenda.
It goes both ways.
It goes both ways.
Yeah.
Lots of stuff developed through the HIV response in antivirals and vaccines, in diagnostics that have been very relevant in other fields, and I think very important work that's been done in other fields, particularly in cancer, that are very relevant to the cure agenda too.
It's great excitement in genetics and gene editing and so forth. We just talked with Christopher about that. So it's exciting. I'm hopeful that we can have another talk with you another 6 to 12 months and see whether we've gone where we thought we were going.
Yeah, of course. You never know. There's always, good news and bad news. Let's hope we have lots of good news.
Exactly.
All right.
Thank you very much,
Thank
Sharon.
you.
Yeah.